So the practical what we did. Remember our first thing, in practical, what temperature is incubated? Anybody? That's great. So you will be handling that particular practical. We tell you what to observe, how to observe. All right. So that is called picture.
Thank you. Perfect. Hello, hello. Can you hear me? Perfect. Okay, hello, everyone. I think I have a very important task, is to keep you awake after lunch. Well, it's quite sad because it's the last lecture, I think. I hope it's also sad for you. For me, it is. So today, I will be talking about I will continue talking about microbiome technologies, an analysis, what we call it for. And this is what we are gonna try to answer today. So we're gonna try to answer one of the two of the three questions. So what are they doing? How are they doing? And for that, I have to tell you that we are gonna be using not only metagenomics and metatranscriptomics, but to do that, we need to also use other metaomics like metaproteomics, metabolomics, and so on. Okay? So we are here in the program. So I made a huge square here because I think we will be covering most of it. So remember, we started here isolating bacteria from the rhizosphere. We have our cultures already showing some antagonistic activities in plate, I hope. We will see it later. Also, we did quite a lot of detailed analysis of 16S amplicon. Yeah? And then now we are moving more into the functions. So how can we see the functions that are happening in our samples, okay, in our treatments. So for that, remember we have 16S amplicon where we look only a fragment, fragment that is conserved among all bacteria, and then we annotate, we trim, we do the quality check, and then at the end we get the profile of the microbiome composition in that particular environment. So now we are going to do shotgun metagenomics. Okay, so we are going to sequence everything that is in that particular environment that you are working with. Yes? So how do we do? Well, we randomly break up the DNA, we sequence all fragments, and then we go for the next step, that is studying the potential gene functions and assemble genomes and so on. So we are going to be looking at that today. This is the general overview. So we get our fragments, and then we do assembly. We know already what is an assembly, how to do assemblies, and so on. And then at the end of the day, we get our metagenome assembled. Okay? Good. Some definitions. I'm not gonna go into too much into detail because limitation of time. Today we are gonna have quite a lot of things to do. But yeah, here you have the definition of metagenomics, that it's basically the study of microbial communities in their original living places. Okay, a metagenome sequence refers to the sequencing of the entire genomes of all microbes present in a sample, okay? Metatranscriptomics, we look at the expression. In that particular moment, we see what those microbes are expressing. Metabolites, we look at the molecules, and proteomics, we look at the proteins, okay? And together with all of these omics, these are what I call it our 4 main purposes or our main objectives. We are looking into the unculturable, remember the dark matter. So here is where we are looking at that. And very important, we are looking in a natural state. So we are not modifying that environment, and we are basically looking at the ecology of those microorganisms in their living environment. But also we can take it to the lab and do comparisons. Like Jos have shown today, we can take all those systems to the lab and, for example, compare health and disease, yeah, development, and so on. Good. I always like to do this, to put you some troubleshooting. I think we already saw most of the problems that we could have when we do, in general, next generation sequencing. But maybe you can help me to fill up here what can affect our results on our microbiome community analysis. Yeah, when you do a microbiome, what can affect our results? Anyone? I cannot hear. Do you hear me? Yes. Is it good? That's a good one. I'm not sure if it's in my list, but yeah, that's an excellent one. Yeah. Yeah, well, for example, sampling method. We already saw it. Yeah, remember when we were talking about 16S amplicon, the sampling method that we use is important. Imagine, indeed, if we look at endophytes, most likely we will have contamination from the host, that is the plant. So we need to take that into account. Yeah? The experimental design, also extremely important. Sequencing strategy. So if you want to know only the taxonomy, then don't do metagenomics. Do 16S amplicon, will be cheaper. Yes? Then we go into the analysis. Yeah, you also need to know a bit about the analytical methods that are out there, the analytical tools, and then we will be able to extract as much information as possible. So this is also a quite important step. And just a little tip, if you have the time and the money, well, it's always good to do pilot experiments. And if you have a bioinformatic expert close to you, ask for recommendations because they will be able to help you in your experimental design. Okay? Good. This is the general overview of what we do when we do metagenomics. So here I took this example from, I think it was a skin microbiome. So what we do is, well, sample preparation, whatever method you use. Yeah, they have already looked at the previous slide, and then they know which method they had to use. Okay, I don't know. Then you do your DNA, but also you can do RNA extraction, and then you will see, yeah, the expression of those genes in that particular environment. Then next is fragmentation. You need to shear the genome, the metagenome, and then you fragment all the small pieces. And then later on, we know that very well from our previous lectures, you do the assembly and then functional annotation. Yes? Good? Okay. How do we do the analysis? Because now we have our metagenome. We have, what do we get when we sequence? What do the companies send to us? Raw data, and the raw data is FASTQ files that contains the quality. I'm not going to go into detail with the FASTQ. Luisa will show you the different steps for the metagenomic analysis this afternoon, but I am going to show you some of the details that you might be interested. So, well, there are several workflows out there. There are many. Yeah, I mean, not only a few. There are many. Or at least I had experience with these three. Not that much with this one, but I know that it's also quite good. Now, I think in my lab and in Jo's lab, we are mainly using Squeeze Meta. And Luisa will give you an example today of Squeeze Meta data analysis. Okay? Good. Just to remind you, remember, the most important is, of course, your research question. When you are doing the analysis of your microbiome, you need to know exactly what you need to ask to the data. But you have to, or you need to, annotate with general tools to know the general functions of your that are carried out by your microbiome. And here, for example, I put you as an example. There are many tools, but this one is the one that we are using now, that is eggnog, that contains most of the annotations already. And then also, it's important, well, to focus your analysis. So once you know more or less what are the functions that are carried out in your microbiome, you need to do a specialized annotation. And in my case, if there is time later on, I will give you an example. We focus mainly in carbohydrate degrading enzymes. So that's the Cassiopeia or the DBCAN database. And also anti-SMAS. We have been talking last week about secondary metabolism. So this is the tool. Okay? Good. So once we annotate, yeah, what we get is taxonomic affiliation and functions. Yes, what do you do with this? Well, alpha diversity, that we already look at it, and I also mentioned that you can do functional diversity. So you can measure the diversity of your functions in your particular example. Principal component analysis, and you can also look at the taxonomic composition. But then, well, you can do many other things with your microbiome data. So going from, well, co-occurrence networks to similarity networks that we were talking a bit this morning, to phylogenetic analysis, and so on, so on. Okay, so there are so many possibilities, but yeah, you need to have the right question to your microbiome. Okay, are there questions? No questions. Okay, good. So today I wanted to focus a bit more on, well, this question. So can we reconstruct genomes from metagenomes? Yes? Yes. So we can do. So that's the process called binning. So in metagenomics, binning is the process of grouping reads or contigs and assigning them to an individual genome. So if you look here on the right, we have this diagram where you have your... Now? No? There was another micro, no? Hello? Yeah? Do we have more batteries? Hello, hello? 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 Well, then I will be close to here. I will try my best. Can you hear me? Yeah. Okay, I will do my best to stay close to here. Okay, so here we have this diagram where you have all the genomes that are in your sample, in your particular environment. So then what we do is we we sequence them, and then we generate contigs. Yeah, you all know how to perform an assembly, and then we do what we call binning, right? Binning is the generation of bins, and we can call it as well metagenome assembled genomes. Okay, so are genomes that are made from metagenomic reads. So we can differentiate two main methods for binning or to generate metagenome assembled metagenome assembled genomes. So one of them is the supervised approach that relies on non-reference genomes and use homology or sequence composition similarity for binning. Okay, so basically you have a reference genome and then you map the reads to or the contigs to those genomes. And then we can have also the unsupervised approach where you don't need to have a reference genome, but it relies more on sequence composition and or in species abundance. Okay, so we are going to see a few examples now, and then it will be much more clear now. So a bit more of information about binning. So we are going to be talking about mainly unsupervised approach, where we need to rely on the characteristics of the sequence composition. So what are these parameters? Well, we have several parameters that we need to look when we are doing binning. So the first one is the read coverage. So when we have a contig, most of the times, the reads that are mapping back to the contig, they always have the same abundance. And we will see in a bit an example. We also look at the chimer frequency. We know what are chimers already. So here we are doing tetranucleotide chimers. Is ready? Hello? 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 No? No. It's okay. Okay, so yeah, so as I was saying, we all know what are chimers. So then here we are looking at chimer frequency. And to do that, we do tetranucleotides. So basically we create overlapping fragments of 4 nucleotides, and then we find patterns that are specific for every species. Okay, we can look also at genome content, codon usage, and other, for example, taxonomic assignment. Let's look at an example. So here we have, for example, MetaBat. MetaBat is a well-known tool for binning. What it does? Well, so this is the normal. So you process your metagenome. Yeah, you get your libraries, you sequence everything, and then at the end of the day, what you get is a de novo assembly of your metagenome. So now what MetaBat does, it calculates the tetranucleotide frequency in your contigs. And then this is matched with the abundance, yeah, of each contig in the library. Then it creates this kind of heat maps where you have in one axis, you have the tetranucleotide frequency, and in the other one, the abundance. And then it compares them. And it gives you the probability that these contigs will be together in a single bin, okay? I'm not gonna go into detail here, but basically at the end, what you get is a list of genomes with high probability to, well, be a genome, a draft genome. Here is an example. I hope you can see. I don't want you to see the table. This is just for your information. These are all the methods that are out there for binning. There are many, many tools that can do this kind of analysis. But here, I just wanted to show you this simple example where you have in the x-axis the GC content, and here you have the coverage, so the amount of reads that are mapping back to that specific contig. Okay, and in colors and in these bubbles are represented the different contigs. The bubble is a contig, and then the color it's the taxonomy. So you can see that, for example, here with a coverage of 140, you have this yellow, these three beans. These three beans have this around 49% GC content. So most likely, the binner or the method to do the binning will take this as the same bean, so the same genome. We'll group them together, and it will give you a genome. Same for the red, same for the green, same for the blue, and the same for the light blue. Is that clear? I don't see you with this light. Yeah, good. Okay, this is a real life example. Okay, this is how looks like when you do binning, for example. I'm not sure which tool did they use here, but what I'm sure is that they use the same GC content and coverage. So then in this particular microbiome or metagenome, you can look more in detail in the paper. The main bacteria that was present was Escherichia coli. It was a gut microbiome. And you can see every dot that you see here, it's a contig, and it's colored by the taxonomic composition. And then all of these dots, they are belonging to the same bean, okay? And we have other bacteria here, like for example, Clostridium or Streptococcus or this other one. Okay, what can go wrong here? Can anyone guess? Yeah. Sorry? Sorry? Exactly, exactly. So when you are doing binning, oh, I should go there. Sorry. This is exercise. It's good. So when you are doing binning, what can happen is that, well, you will not be able to differentiate, for example, between contigs from the same species or from the same, sometimes even genera will be mixed up. So this is a bit risky, okay? But nevertheless, we have many tools that can refine them. So we are gonna see a few examples. Oh, that's great. Oof. Yeah, thank you. I think it was reflecting in my glasses as well. Thank you. So then, for example, let's look here. So we have this bean in this pie chart, as you can see, and then we have Pseudomonas mainly, and few reeds or contigs, yeah, contigs from Enterobacter. But here, the problem is a bit bigger because we have Pseudomonas as well, but we have several species of Pseudomonas. To differentiate between them is going to be quite complex. Well, but we have something to use for this. We have tools, and Luis already introduced them, one of them, that is CheckM. You remember that from one of the first lectures last week? I think you do. Well, CheckM look at the presence of markers. It checks for the presence of 43 markers that are present in all genomes in single copy. And then you can evaluate how complete are your genomes. And for example, well, I'm not going to go into detail here because we have more examples further, but we can evaluate the completeness, the contamination, but also the strain heterogeneity. And we are going to go deep in these two parameters. Is it clear for all? So the first one. So we have the contamination, yeah. So the contamination and the completeness. So we are gonna check for single copy marker genes in all our beans. So we are going to be searching for those 43 genes, yeah? And then we are going to determine if those 43 genes are present in all our beans. And that's going to give us a kind of an estimation of how contaminated are our beans, or maybe they are not contaminated. But here I bring you an example. This is the reference. So we have here 10 genes. You can see that we have 10 genes. And this is the new genome. So the bean that we have generated using our programs. So we have 10 single copy marker genes. So, and here, when we look at our new genome, we see that one of the genes is missing. And this one is repeated. Yeah? So that's going to give us this result. So 9 out of 10 genes are present and 1 gene occurs twice. So that gives us a result of 90% completeness and 10% contamination. Yes? Is it clear for everyone? So imagine doing this for 43 single copy gene markers, yeah, in all your beans. This is what we do during binning. Yes? Good. Then let's check this parameter, the strain heterogeneity. So this indicates the source of contamination. So for example, this will tell you if there are other strains of the same species versus distant taxa. I think it's quite interesting, this parameter, and we should take into account when we are looking at our beans. How many of you are working with metagenomes? Well, quite a few. Are you planning to do binning? Yes, it's cool, eh? Okay, so when we look at this, for example, we have this bean that has 85% completeness, 15% contamination, and 100% strain heterogeneity. Then the other one has 85% completeness, 15% contamination, and 0% heterogeneity. So this one, for example, most likely are going to be different strains from the same species. And this other one, most likely, is going to be a strain from different species. And if we go back to the example, this is what we see here. Well, here, for example, the result will be 100% heterogeneity because they are from, well, not from the same species, but they are from the same genera. Okay, and in this case, they are from the same, from different genera. So it will be easier for us to clean up this one than this other one. Was this clear for everybody? Good. So these are some of the tools available for binning, for the cleanup of the binning. So for those ones that are well, doing metagenomics and doing the binning now, these tools we have used in the past in some of my projects or in my group now. Okay, so, but as I said, there are many tools that can do this. So, and well, when you are working with metagenomics, you want to extract as much information as possible. And as I told you, the binning tools are using different methods. So what if we could have a tool that could embrace all of these methods together and give you a final bin, a consensus bin, yeah, a clear result. So that's what it's doing, that tool. And I'm giving you this example. So if you want to get more, you can look at this website, Meren Lab. It's quite nice. It gives you, well, quite a lot of examples and very well documented and organized. So imagine that we run our metagenome and then we get one bin, only one metagenome assembled genome. in this case produced by Concord, is this one here. Then we run another tool that is MaxBean. And then with MaxBean, we get, instead of one bean, we get 1, 2, and a few small ones. Yes? Then we go and then we run another tool, because every tool is taking different parameters into account to build beans. So then this other one gives another different result. So it gives you one bean here, another bean here, another bean here, and then a few more beans here. So what that tool is going to do with this is going to take the output of all of these tools, all of these binning methods, and it's going to merge in one. So then at the end, that tool gave one bean, two bean, and some small ones. So it's kind of refining the result using as a base different methods. So for those doing metagenomics, I think this is going to be quite nice, right? Yeah. Can you see this? Well, it's not that important. Well, it's important. So this is how do we check for the quality of the standards, quality assessment standards of beans. So for that, in 2017, Bowers et al. Published this nice perspective in Nature Biotechnology, where they look at the minimum information about metagenome assembled genomes. And then here, what they did is kind of, they settled the standards for metagenomes, for metagenome assembled genomes, so for beans. And for example, to have a finished MAC or a finished bean, what you need to have is all single contiguous sequence without gaps. So basically 100% correct genome. So if you have a high quality draft genome, to have a high quality draft genome, you need to have, maximum 90% completeness, then 5%, less than 5% contamination, and so on, so on. You have medium quality and low quality. Yes? Okay, so this is one of my last slides before the example. So then, yeah, what I wanted to emphasize with this last slide is that when we do microbiome data analysis, we need to take into account not only sequencing data if we want to draw conclusions about it. So we need to have, well, metaproteomics, metabolomics, and then integrate all of it together, to get the answer to your questions. Okay, I don't know if I have time to give the example, the endophyte microbiome. Yes? Then, then I will summarize later. Okay. So you have been already, just already talked about this, about the disease suppressive soils. You all know what are disease suppressive soils, so I'm gonna pass this fast. But this is our study system. So when I was doing the postdoc at Josh Group, this is our system. We work with sugar beet plants and with the damping of disease caused by Rhizoctonia solani. So, and here, it's not the first time that you see these two pictures. We have conducive soils and suppressive soils, plants in suppressive soils and healthy and happy. As you can see, they grow quite well. And then in conducive soils, when you are the pathogen, they die. So in Josh group and in other groups, we have been looking at different meta-analysis and functional analysis to determine which microbes and which functions are involved in this phenomenon. So basically, it's the who, where, and what. And that has been mainly done in the rhizosphere and never in the endosphere. So our question here was, what is the role of the endophytic microbiome, so bacteria living inside the root system, in the protection of plants in disease suppressive soils? Yes? So that was our main question. So this was the experimental setup that we follow for this work. So we have plants growing in conducive soil, plants growing in suppressive soil, and then plants growing in conducive and suppressive soil inoculated with the pathogen, in this case, Rhizoctonia solani. What did we do next? Well, we harvest the roots, and then we root surface sterilize the root system, and we blended them. So we did root maceration to make a kind of sugar beet juice. So, and then using this, as your colleague mentioned already, this is quite risky because you will basically get reeds coming from the plant because it's endophytic microbiome, and then you are grinding everything. So what we did here, we did a nicodens gradient separation. So maybe it's a good method for you, for the ones working with endophytes. This will improve your sequencing output. So what it does is, So you create different gradients going from 50, 35, 20, and 10. And then you do a differential gradient centrifugation at high speed. And what you will get is that the plant cells that are bigger goes to the bottom, and then the bacterial cells stays in the middle. And then with a Pasteur pipette, you can fish your microbiome. So that's basically what we did. So with this sample of microbiome from the endophytic compartment, from the different treatments, the different samples that we had, so what we did is a culture-independent method where I did metagenomics. So basically, we sequenced everything that was in there. And then the other approach was a culture-dependent approach. Remember the first day that we were talking about culture-dependent methods. So here I did the same, dilution plating and isolation of bacteria, like you did in the practical. Okay? Good. So then you get your metagenome, and then, yeah, you get quite a lot of reads. You need to analyze. Well, thanks to these guys that are called bioinformaticians, I was having a good time with them, and then they teach me quite a lot. So I really have to acknowledge them because I'm not bioinformatician by training, but I really learned a lot from them. So we followed several tools and methods to get as much information as we could from the metagenome. And well, just the first snapshot that I want to show you is that, yeah, 75% of the reads from that metagenome were belonging to plant. So only 25% were belonging to the microbiome. So this method that we use here, yeah, was quite, was okay. So we got rid of quite a lot of plant, but still there was a lot of plant read in the final result. So there is no perfect method for this. But if we don't do this step, then we will have get 99% plant and 1% bacteria. At this moment, I became vegetarian, and because I was hating plants. So because, yeah, imagine that was a lot of money that we throw it into the bin. Anyway, so we also had to do binning, and I can tell you from this, the amount of read that we got, we were able to get, I think it was at the end, 24 high quality beans that were used further on in the analysis. But I will not go in detail in this part. It's not the first time that you see this kind of plot. Last week we saw it as an example of 16S amplicon analysis, yeah, for differential abundance. You can use the same for metagenomics. So as well, you can see in yellow, I'm showing you the bacteria that were equally abundant in healthy and diseased plants. And then in blue, for example, are the ones that were enriched in suppressive soil. So what is quite striking is that, well, mainly Bacteroidetes and some gamma protobacteria were responding somehow to the pathogen infection were more abundant, because remember, this is metagenomics. We can talk about this is more abundant, but we don't know if they were more expressed or not. Okay? Good. You also saw this before. I show you in the annotation tools. So this is the way that we look for the functional genes that could be involved in this disease suppression. As I showed you, well, mainly we had bacteroidetes that were highly abundant in this system. So then here, what we did, because think in this way, so we are infecting sugar beet plants with Rhizoctonia solani. It's a fungus. So if there is something going on, most likely it's that the bacteria is degrading the fungal cell walls, for example. That could be one of the possible results, right? So what we did was to look at carbohydrate degrading enzymes. For that, we use a specialized database like DBCAN. Yes. So with DBCAN, we were able to identify the genes that were involved in this response in a bit more specific way, because here we are looking at the ones that are able to degrade carbohydrates. So how to read this? Well, this is a network analysis based on HMM domains. We also know what are HMM domains. And then each of the nodes that you see is one gene from the DBCAN database from the endophytic microbiome. Then the H's is the identity between the different genes, the different nodes. Okay, so then basically this is a kind of a measure of the distance between the different genes. So, and then in colors, we have the different types of carbohydrate degrading enzymes. And then in squares, we are representing the ones that were enriched in the suppressive soils, in the plants when they were in the disease suppressive soils growing, and then we isolated the endophytic microbiome from there. So as you can see, most of the chitinases, yeah, you can see a few somewhere here, yeah, many chitinases were significantly more represented, more abundant in the disease suppressive condition. So, and when we look at the functional annotation of those chitinases, they were belonging mainly to the Chitinophagaceae family. Okay, do you remember where Chitinophaga was before? So in the taxonomic analysis, we already found that Chitinophaga was highly abundant. Yes, good. You also saw this before. So this is the output of anti-smash. Yes, good. So in the endophytic microbiome, we were able to predict 730 biosynthetic gene clusters. So normally in a genome, you get around 20 max, 15, 20, 25, depends how big is the genome. But these are all the, all the biosynthetic gene classes that we got from the whole microbiome of the endophytic community. And they had all of these affiliations, so these, well, NRPS, PKS, bacteriocins, all kind of compounds. We can look one by one. I show you already this, and then see, for example, the structure or the gene structure as well, and so on, how the genes are organized. But we can look also this in a network analysis, and I will go, if there is time at the end of the day, I will show you how to build this network. Yes, good. So then how do you read this? Well, each of the nodes, it's representing one biosynthetic gene cluster. So one group of genes that is involved in the production of secondary metabolites. Then in yellow is the same than the one before. In yellow are the ones that are non-significant. So they were equally represented in disease and healthy. And then in blue, the ones that are overrepresented in the suppressive soil or correlated to suppressive soil. Sorry, this is gone here. So then when you look at this, well, and then the edge here, for those that are more interested into networks, nodes is represented by the gene, and then the edge is representing the distance or the identity between the nodes, okay? Then you can annotate them based on the origin. So if they, for example, are NRPSs, siderophores, aryl polyene, whatever kind of compound, but you can also see the taxonomy. And what was more striking is that, well, in this side, we could see that NRPSs and PKSs were highly abundant in our microbiome, and especially the ones belonging to Flowobacterium, but we will come back to this in a bit. So when we go a bit more into detail, this is what we got. Well, if we look at non-NRPSs that are producing secondary metabolites that have been described in literature as involved in disease suppression or antagonistic activity, this is what we found. So see here we have conducive soil, suppressive, and suppressive inoculated with the pathogen. So we don't see any pattern. So most likely these ones in the endophytic compartment, they are not involved in disease suppression, okay? But then when we look at the unknown NRPSs that were a lot, this is what we got. This nice pattern in this heat map where we see that most of the unknown NRPSs were overrepresented in the endophytic microbiome when the pathogen was present. So that was quite important for us. Is this clear for all of you? Okay, so now our goal was going from microbiome to microbe. So we have done quite a few bioinformatic analysis, and we have squeezed all the information that we had from the endophytic microbiome. So it's the time to go back to the lab and try to see, well, what these microbes are doing in real life. So as I told you, we got from the taxonomic composition of our metagenome, we got Chitinophaga, we got Flowobacterium that were highly abundant in our taxonomic analysis. Then when we look at DBCAN database, so carbohydrate degrading enzymes, we saw that these enzymes, chitinases, were highly abundant as well. And then when we look at natural products, so secondary metabolites and biogenetic gene clusters, we saw that there were a few NRPS and PKS that were highly abundant, okay? So, and remember, I also did a culture collection. Actually, I say here 900, but I had 1,800 isolates. So it was quite a lot. So what we did, we cross-link the bacterial collection that we had plus these functions and taxonomic composition. And then we end up with several bacteria, like, well, the ones that we were looking for, Chitinophaga and Flowobacterium, that contain chitinase genes and 4 NRPS, PKS genes. Okay? So we did this cross-link by PCR. All with me? No questions so far? Good. Okay, so what was next? Well, as I said, going to the lab and going to the plant as well. So what we did was in vivo activity, colonization, and also expression. So we look at the expression to see if indeed these genes were involved in this activity. So here in the y-axis, you have the disease incidence, and in the x-axis, you have the different treatments. So in the first one, you see the disease, well, the control. So plants are getting sick when you add Rhizoctonia solani. So this is what you get. Yeah? So this is a conducive soil where we are not inoculating any bacteria. So when you add Rhizoc, the plant dies. So when we add the 2 bacteria that we selected, Flavobacterium and Chitinophaga, we see a reduction on the disease incidence, around 40%. I mean, it's okay, it's good, but still a lot of disease, right? So, but what happened when we add them together? So what we saw that the reduction was around 20%. Yeah. So you see that indeed when you add these two microbes together in a synthetic community, that just has been talking about it today. So we see a synergistic effect, and these two bacteria help each other and protect the plant against the disease. Then the next question was to determine if the genes that we found, if those genes will be involved in this phenotype that we saw. So for that, we look at the expression, and then we saw, well, here you have different treatments, but what is more important is that here what we see is that one of the biosynthetic gene clusters was highly expressed when the pathogen was present. And then what we also saw is that the chitinases, when the pathogen was present, also the chitinases expression was also quite high. So this is already telling us quite a bit. So what we did next, we took this biosynthetic gene cluster, and then we performed mutants. We did mutagenesis, and then we knock out one of the genes. I think it was the PKS gene. And then we went back to the plant. So we inoculated together with Chitinophaga, and then we saw a similar reduction than when we were adding the individual ones. But then when we add Flavobacterium alone, the disease incidence went up almost at the same level than when the bacteria was not present in the control. Was it clear? Good. You already saw this in your talk. I think I can say that this was work for how many years? 6? No, but I mean the whole 12? More, I think. Okay. Well, this is a lot of work from different postdocs and PhDs working in Josh's group. And what we think is that, well, how we think it's working is that there are 3 barriers. So the first barrier is the rhizosphere, where you have many different compounds, many different taxa that are helping the plant to be protected against pathogens. Then, of course, the second layer of protection, or the second barrier, is the plant, the immune system of the plant, and the physical barrier. And then there is another barrier that is the endophytic compartment, the endophytic bacteria that lives inside the plant. So how do we think that it's working? Well, we think that, yeah, Rhizoctonia tries to enter, then chitin is released or break down by the plant or by the bacteria. We don't know. Then Chitinophaga will degrade these fragments of chitin, and these chito-oligosaccharides, what we think is that they are activating Flavobacterium. And this, Flavobacterium then produce these secondary metabolites that inhibits the growth of Rhizoctonia solani. This is how we think is working. And well, now we have a PhD working on this project together with Josh and me, Sinia, and she's focusing mainly in this part. Yeah. Okay, so this was the end of my presentation. I wanted to show you an example of of microbiome data analysis, of metagenome analysis, how you can go deep to the function. So I also prepared a summarizing slide for the 16S and the metagenome amplicon. I'm gonna go back. Sorry? Sure. Yeah. No. This is not the big summary. This is 16S and metagenome. Yes? So, okay, so remember, I think this is something that it's important for all of you to, this is the take-home message of all the lectures that, well, at least the ones that I have been doing and many of the things that Jos and Guillermo and Luisa have been telling you as well. So Amplicon data, 16S, 18S, and ITS. So this provides a snapshot of the taxonomic diversity, okay? So this only gives you information about taxonomy. There are some ways to get information about functions, but we didn't go in detail here, but there are some ways to get into there. I put here inexpensive, I mean, it's money, so, but well, it's cheaper than metagenomics. So, and you can get, well, you can sequence quite a lot of amount of samples for less money. It works well in low biomass samples and samples with high amount of concentration. So you saw that during our DNA extraction with much DNA. So, but this 16S amplicon needs in the range of picograms. Okay, so you can really do it with low amounts. So it's not good for strain identification, and well, we have seen already the problems of this. So we all know what it is. So it can be biased by the primer choice. And well, we have to also see the technical artifacts. Remember when we were looking at data2, for example, or OTU generation. Shotgun metagenomics. So shotgun the metagenomics can generate taxonomy, yeah. So you can generate taxonomic profiles, not only using the 16S from the metagenome, but also using other housekeeping genes that are conserved, okay? This is an advantage of doing metagenomics. You can get not only the taxonomic profile, you get also the functional profile. This can go a bit further, and then you can get strain-specific taxonomy. Problem, of course, there is always a problem. It's expensive, yeah. So it's quite expensive as compared with 16S amplicon, and well, you have to see if it's worth it for your research question. And something else is that, yeah, as someone mentioned over there, you need to be very careful with the host DNA content, because, well, as I show you, 75% of the reads in my endophytic microbiome were belonging to the plant. So we almost sequenced the sugar beet genome, and we didn't want that. We only wanted the microbial community composition. Okay, I think I'm done. So I think I will give the floor to Luisa and then later questions. Yeah, good. Yeah, we need to change the laptop as well, right? No, no. Ah, mierda, sí. Es verdad. Espérate. Espérate. No, 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 no. Vamos a ver. ¿Tú lo has conectado antes? Ostras, pues entonces Guillermo lo tiene. No se puede, Luisa. No se puede. Pues se la tienes que pasar. O me la pueden mandar al mío, sí. No, al otro. uv.j. Sí. Ah, no tengo yo internet, tío, ahora. ¿Cómo era? R bala. Oh, ya. ¿Tú me has conectado tu teléfono ahí entonces? ¿Tu portátil no lo ves? No, necesitamos un pendrive, Luis.
Ya, yo no tengo. Em, tienes suerte. Qué suerte tengo. Hay un problema, no pasa nada.
Yeah. Oh, I got one. Ready.
Basta. Okay, so let's get started. This is the last, yeah? A sound. I think this is fine. No? Still not.
Just stop this way. That's also online. What do you? This one is also motion.
You're saying that I should use this instead of that one.
No, they're saying this one has the noise.
Ah,
So we bunch of
Hi. Better like this? No. No. How about now? Yeah, I can hear it, but I don't know. Hello? Better? No. Better now? Oh. I can yell. Okay. Bye. And what if I use another mic? It's not because it was the speaker, right? Hi. Is this okay? No? Hi. Better? Yeah, but they said no. One. This is option one. Option two. One? Is it better like this? Oh. Hi. 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 I can I can be loud. So hello. Yeah? Now it's good? It's not. Okay. And if I speak like this, can you hear me? Maybe we just go without mic? But I have this one.
Okay, I think the remote will work here, right? And then we turn off the sun. Okay?
Ah, so no mic from the. But I would be facing. I would be giving them my back if I stand in the middle. But it was okay for Victor. What changed? This one? Hi. It's it works. Yeah, I cannot hear it from here. Hello. Hi. Still there? Yes. Would it be an option then for them to do the practical over there while we fix this? Oh. Hi. Is it fixed? Fixed. You can still hear it. So we just proceed like this then. Okay. All right. Thank you very much for all you did. Okay. Are you hearing me? Yes. Yeah, I think so. Yes. Okay. So, metagenomics practical. I'm gonna show you an example more focused on the outputs and the practical things compared to Victor. And this is our last practical. And in a way, having metagenomics as the last topic is actually ideal because you're going to see many familiar things, hopefully. Because this is like mixing a lot of things together. So you will see. So in our diagram, I only highlighted this part, functions and meta-omics, because that's what I'm going to highlight in here. And this is the data set for this practical. So my example will be based on this. So we have a substrate that we add to soil, and then we sow the plant after 3 weeks. We can remove shoots and roots, collect rhizosphere and phyllosphere, extract the DNA, establish glycerol stocks, and then send everything for shotgun metagenomic sequencing. And we have all of this, just a control, and then for the substrate, we have 4 different concentrations. And I want to highlight this part, which might seem like a little detail, establish the glycerol stocks, but that step is the only one that will allow you to come back to your samples after the sequencing has been done. So it's always important to establish and have a collection of your samples. Even if your initial plan is not to sequence again, you never know what can happen. So you should have that reserved there. And I'm going to talk to you about squeeze meta. So as Victor said, this is the one that we use the most. And the reason why is because it's flexible, but it's also very intuitive, it's well documented. So if you go to the GitHub page, I have another slide with it a little bit more visible. It's very organized. The creators were very organized in the outputs. Even though there are so many, you're not going to feel lost. So that's why we prefer this one, because not only the bioinformaticians use it, but also other PhDs who just happen to have metagenomes, and they are able to use this very comfortably. So that's why we choose it. And I'm going to show you briefly the diagram or the workflow that they follow. So you start with your DNA reads, you send for sequencing, and as you remembered with Victor today, you get FASTQ files. For both metagenome and metatranscriptome, it will be the same format. And then after you have your reads, you learned before at the beginning of this course that you should always check the quality of your reads. And we did it with FastQC. I hope you remember that one, because we're gonna skip this step today. We have seen it like 2 or 3 times already. So I'm just gonna assume that we all know it and that we start with an assembly. So after you have done the quality check, you're free to go and assemble, do a huge assembly for your metagenome. And that you can do with MegaHit or MetaSpades. So Spades has a version for genomes. This MetaSpades is for metagenome. And then once you have your assembly, it's also important to check the quality. This we also saw in the H3, I believe. And we use Checkm and Quast. I hope you remember. So you see, metagenome is very much like a summary in itself of the whole course that we've been doing for now. And after you see that your assembly is okay, you can proceed with gene prediction. And for that, they use Prodigal, which Guillermo mentioned some days ago. And then you can do the functional and taxonomic annotation. Squeeze Meta has KEGG, COG, and PFAM as databases used for this, even though there are more and you have seen them. These are the options available in Squeeze Meta. And after that, you can get the count or the actual abundance of the genes and the functions because you can map these reads, the initial reads that you had, to the contigs. Now, since you already assembled, you have more contiguous sequences and it's more complete information. And you have annotation already, so you can count those reads and have as a result the abundance of each annotation that you have done in your samples. And with that, we can generate these gene and contig tables that you will see in a couple slides in the future. And then after that, from the assembly as well, you can do binning, which Victor talked about very nicely, and you can get the metagenome assembled genomes. And for this, Squeeze Meta uses MetaBat. They also have the option of using MaxBin, and then DAS tool is always the last option because they use it as a refinement method for the other binners that they used before. So that is our workflow. And this is how we're gonna look at it. So first is quality check. We're not gonna stop there too long because we have seen that before. Second, third, fourth, and then all the way to binning. So these are gonna be the steps that we see today. So first we have the read quality check. This is a slide that Victor showed. You can see the quality of your base calling, and we use FastQC in the examples that we saw the days before. And then we have the assembly and the quality check of that assembly. And I hope you remember this table. This is the same table I showed on day 3 for genome, which is the statistics table that Quast gives you. And this is exactly the same one. And when you do it with a metagenome, you get the same format, but the numbers change quite a bit. So I hope you can read it. Otherwise, I will just read it for you. So this is the largest context. For genome, it's like 2 million. And for metagenome, it's less than 500K. And then the M50, which I hope you remember, is way larger for genome than for metagenome, which means that here we have way larger context in general. Not only here the largest one, but the M50, and in metagenome, they tend to be shorter. And then if we check these values, on the other hand, metagenome will have them higher. So the number of contexts, here there are a lot of contexts, which makes sense because you not only have one genome, you have all the genomes that were in your samples. And here we only have 10 contexts, only one genome. And then the total length is also dramatically different. So in here is huge, and it all makes sense. So you have way more information, but you also have more context. And the reason is that the metagenome is highly fragmented. So you have more info, but since this is shotgun metagenomics and you're just sequencing everything, it tends to be more fragmented and shorter context is your starting point. So those are gonna be the differences in your numbers in this assembly quality check. And then we can continue with gene prediction. This one includes the 16S, so you're also gonna have this information. And what you get from Squeeze Meta, they use Barnap for this, which I am sure you heard here before. I think Guillermo mentioned it. And this is a FASTA file, so it has the IDs of the context. In this example, they used megahits as assembler, and then the sequence of it. And in here you have 16S, a 16S, and this one is a 5S because, well, it includes 16S, but it's not limited to that. But in case you want to have the 16S, you can simply filter the files that you get and just end up with one that only has the 16S, and even the taxonomy is already assigned to it. So I have a question for you. Since we have 16S information in here from our metagenome, do you think we can make a phylogeny with them, with the 16S, like we did in the days before? Okay, so the answer is yes, but no. So yes, we can, but not as it is. We have to do some extra things. The reason why is what I said before. It's fragmented. We don't have primers. We're not targeting a specific region of the 16S. So what we get here are many different regions of the 16S that we don't even know because the sequencing was done randomly. So what we can do to be able to do the alignment for the phylogeny is go to the Silva database or some other database of your preference, and then map these reads that we got against the Silva database, because this we know has the full 16S regions. And then once we do that, with those, we can do the phylogeny. So yes, we can. Then we have the annotation. So as I said, the options for Squeeze Meta are KEGG, COG, PFAM, and of course the taxonomy. So you're gonna get a huge table, way bigger than this. It's in the last data I processed with this, I ended up having hundreds of thousands of genes. And then you get your genes, but also the ID and the description and function for COG, KEGG, and PFAM. And then in the end, also your taxonomy. So you get already that. And then you can get a count. So if you map your, as I said before, if you map this were the original samples that we had. This is only a subset of 5. the 5 replicates of the highest concentration that I had. And then you can map the reads, the shorter ones that you had at the beginning, against the contigs that you have now because you already did the assembly. So what they do in Squeeze Meta is that they count all the reads that you had in the sample, and then they count how many of those mapped to the contigs, to one contig at least that you had. And then it gives you the percentage of mapping, which is in here. It looks very good in here, and they also count the total bases. So actually, the mapping percentage is also a very nice indicator of the quality of your data, of your process, let's say, of your data analysis. So you can have a high mapping percentage or a low mapping percentage. High is considered above 70%. So this example is quite nice. But if you have low mapping, you shouldn't be too worried about that depending on the nature of your data, because this is even to be expected if you have complex samples, so soil or gut samples. If you have too many microbes that are uncharacterized or rare, this can also happen, or if your sequencing depth is low. So don't get scared if you have a mapping that is below 70. This actually happens a lot to me. Oh, this is because we have soil samples a lot of the time. Okay, it is working, just not the thing. And then you can get the gene and contig tables, where you have in the first column to the left, you have the open reading frames. So every single gene that was predicted has an ID. And then for it, you have the taxonomic assignment in the second column, then the functional annotation out of all those databases that I mentioned, and account information, because now that you mapped the reads to the contigs, you have counts. So each column over there belongs to one sample. And you can also see that the format of that count table is very similar to an OTU table, many zeros. So this is also compositional, and you have to keep that in mind for the rest of your analysis. So now that we have all this information, taxonomy, function, and even counts, it all looks integrated and it looks like something that you can already use for your analysis. So how can I use that for my experimenting? Here you have to bring back your own experimental design. Is easy to get lost in all the data and all the numbers, but you have to remember your question, because otherwise with all this information, you can just drift away in the data. And in this example, I'm just gonna focus in the highest concentration that I had, so the 100. And you can do plots already to explore this very dense data. So please remember that you first have to correct for compositionality and sparsity and dynamic range, just like you did in the other type of analysis that we saw in the previous days, because this is still compositional data. And for example, what I did was a PCOA. So you can see that my 100 concentration, which is over here, separates very nicely from the rest. And even in a heat map, I really like heat maps when it comes to metagenome, because they can fit so many. In here, I think I have like 3,000 features, and it doesn't look too crowded. If you try to do a bar plot, that's going to be different. So for starters, I really like a heat map. And you can already see a pattern over here. I'm sorry I'm pointing like this. It's not working. Of overexpressed or overpresent features over there. And there's also this one, where it's actually losing the features. And then based on this general approach, you can try to filter a little bit or focus more your analysis. For example, I could say, okay, that corner over there with the 100 concentration looks very interesting to me. So I'm just gonna cut and extract those and keep my analysis with that. Because if I always use all my data for the rest of the steps, it's gonna be very, very consuming in resources, because this is huge data sets that we're talking about. And then let's not forget the binning. So this is the last step that I want to talk to about in respect to the workflow of Squeeze Meta. So the binning gives you 2 files per bin in Squeeze Meta. They can be a lot. In this example, I had 500 bins. And what you get from it is a FASTA file with the name of your bin, which is 1. They didn't think too much about them. They just number them. You have a FASTA file and then a taxonomy file, because they assign a taxonomy to the bin that is possibly a genome that you can retrieve from your data. And this is how they look, more or less. So you have the FASTA file, each contig that belongs to that bin, plus its sequence, and then the same contigs with the taxonomy that was assigned to them. So they can differ a little bit, but in the end, there's going to be a consensus. So for example, this reached genus level, this one didn't, but it was the same family. So it's very easy for the consensus to just say, yeah, this is a Opergundimonas. And then Squeeze Meta uses CheckM to generate their own kind of table, where they give you the number of bins that you obtained in your analysis and how many of them were complete in a 50%, 70, or 90%. So completeness, remember that Victor already explained it very nicely. It's about the 43 gene markers that CheckM uses for the diagnostics. And then they also give how many of those bins had less than 10% contamination or more than 50%. And in the end, as a summary, they give you the high quality beans, which are more than 90% completeness and less than 10%. I have 0 of those beans. In most of my analysis, I've had 0. I think the highest I've had is 2. And once I was talking with a bioinformatician from NEO that is very experienced in this, and he said, yeah, I don't know who decided that this was the threshold, because it's very rare to get a bin with this quality. But the good quality bins are 51, so I'm happy with my results. So Squeeze Meta gives you this summary table in the end. That is the last thing that they do. And once you have your genomes, which is a product of the binning step, you can do something like this. So in here I have a phylogenetic tree of all the genomes that I retrieved from my samples. This is an example that the authors of Squeeze Meta made, and they did it with ITIL. So what you can see in here is that they have the bins over there, and in the outer rings, they have over here all the samples that they had, and in red, the abundance, so how many counts of each bin were linked to each sample, and then the completeness based on the CheckM value that you get. So after I did all this and Squeeze Meta is done and I have all these tables and this data, what can I do? What now? So you can do targeted analysis or untargeted. Untargeted is a little bit what we saw before. So it's like a big picture of everything that you have. And you usually do it as an exploration, or if you want to focus a little bit more your research, then you have to do that first. But then, based on that untargeted analysis, or based on literature, or maybe because your research is the continuation of someone else, you can have a point of reference already, or have an idea of the functions that you're interested in, or the taxa that you're interested in. And so you would go for targeted analysis. So my example for you for targeted analysis is this project where they were interested in an oxalate degrading enzyme, which is in here. So this is the EC number. This is a part only from a pathway in the KEGG website. So they were interested in this enzyme that takes the oxalate and makes it formate, 4112. So since I already know the interest, I can just filter all the tables that I got and just keep that information. Plus, it's also going to save me a lot of time in analysis. And you can do something like this. So this is the enzyme that I was interested in. And X, you have control, but also the concentration that I told you that I was interested in. You can do it with all of them if you want. And then the read count for each of them. But the very nice thing of metagenome, and please remember this, is that you not only get function, you also have the taxonomic information, so you can actually see which taxa are contributing with that function in your system. So in here, I had some Burkholdereales that were very high in my concentration 100, and you can see more about the dynamics and what is actually happening in your system with this kind of analysis. And then we come to metatranscriptomics. So the good news is that they are very, very similar. They are so similar that you can use the same pipeline for that analysis. So Squeeze Meta is an all-in-one pipeline, very convenient, and you can also use it with this. There is only one main difference, which is very, very important, and that is the interpretation. Because meta-transcriptomics that has transcripts. So what you get is not going to be the functions or the bacteria that are present in your system, but the ones that are active, that are being expressed. So that is the main difference. It's a very important one. But then it is also recommended to have metagenome with your metatranscriptome as well, because you could map the transcripts back to the metagenome to get more information from them. And then I want to show you a little bit how you can run Squeeze Meta. So for this, you need a server. There is no question about it. It's too much data. This is very intense. So you need a server, a Linux server to do that. But the good thing is that they are very, very descriptive with everything. You are not going to get lost. And they also, if you have questions, you can also talk to them, contact them via GitHub, and they're quite fast responding. So that's very nice. So this is the GitHub page that you can check for yourselves if you want. And they have this execution. This is an extract from their own GitHub. So this is the command for running Squeeze Meta. It's very simple and it's very short. So you have Squeeze Meta, then you check, you can choose the mode of assembly. So this one, you have to consider when maybe your server is not that big, it doesn't have that much RAM. So they have different modes of assembly depending on how much resources you have available. If you are willing to sacrifice a little bit of quality to be able to do it with less resources or in less time. And then you can just assign a project name, because all the outputs that they're going to give you, which are a lot of files, are going to have that name, so you can always identify them. And then this file is the samples file, which looks something like this. So you can have your samples in another directory, and the only thing that they ask from you is that you make a text file when you put the forward and the reverse read, and you indicate which one is the forward, which one is the reverse, and then the name that that sample is going to have, because that's the one they're going to follow the rest of the analysis. Then over here, you can just put which directory is the one I have my files in, and that is it. You can start running it. It's going to take some time, depending on your resources. It can take up to 2 months with a metagenome that is not that big, maybe 1 month if it's a small one. You need Linux. There's no workaround for that. And that is it. Thank you. So do you have any questions for me? Oh, it was very clear. Wow. No questions? You did say that for the validation of the metatranscriptome, along the metatranscriptome, we should always we should go for the metagenome, right? For the taxonomy. Well, you don't have to. It's just nice to have it. Yeah, yeah, yeah. Yes, it's optional. Yeah. And second is like if we do the meta transcriptome, there are some papers where they say that we have to revalidate the transcriptome results with the RT-PCR. So is there any must to do that? Can you repeat? Imagine we are doing the meta transcriptome and I'm doing the targeted analysis. Okay, so do I need to validate the results by doing the RT-PCR amplification of the targeted gene? Okay, thanks.
Maybe about your first question, you need to have metagenome and metatranscriptome. In fact, the answer is that would be best, okay? Because if you are doing a metatranscriptome, you need to map the reads somewhere. So the best would be to have an assembly of your metagenome to map the but it's like when you have a genome, and then you do the transcriptome of that bacteria. So you map the bits to the genes. With metagenomics, metatranscriptomics, is exactly the same. So of course, you have the money and you can do it. It's better to have the metagenome. If you don't want to do the metagenome, then you can assemble the metatranscriptome as well. But it will be, you will get shorter copies for sure. And then what was the output? What was the output?
As you said that we could go for the beginning and that things that no, we didn't.
I never tried that. Going from metatranscriptome to beam, I guess that you will have, well, you will need long context. How I think I will need to have a look at your specific quest analysis and check-in analysis to see how big are your context and so on. I never did it, but you can give it a try. You know, it's for free. Bioinformatics is almost for free.
Wow.
One thing with 95 to micro, because if you go microtranscript or microgenome or sample, that's very costly. So normally what we do is transcript them, and then we pull the samples of the other ones, you know, of the references together, and that we do one run for microgenome. Because you only needed to map. So you don't need to have each reference. You basically make a proposal and map. Also depends a lot on how diverse is your environment. Is soil, yeah, it's quite high level. And the other question, yeah, the question is if we need to revalidate the analysis of the results obtained with the metatranscriptome by qPCR. Yeah, some reviewers will ask for it, some not. I think it's always good to double check my qPCR that you were getting the metatranscriptome if what it is. Yeah.
One part that you showed where you have shown the tree count, right? Yes. So when we make this is very basic. So do we need to make the graph with the normalized data or with the non-normalized standalone data? No, you should always normalize. Always the normalized. It means you are access so much to the large like 50 objects or something like this.
I don't know exactly which graph was it, but yeah, so whenever you are doing a graph for a publication, do normalization, because you need to take into account the total number of reads, well, what we have been talking, compositionality, sparsity, it's always better to normalize.
This is just to confirm that for the carbohydrates, why are we specifically going to the
Yeah, that's an excellent question. So you can go also with a cake, but the good thing of this scan is that it's manually curated. So many of these tools that are specific or specialized in one process, they have been manually created by a group of people, like for example, Antismash. Antismash, the latest version of Antismash, we were a group of 60, 70 people that we were manually entering all the bacteria being clustered by hand. We go to a paper and then we see which cluster it is, we see which bacteria, and then we put it in the data. Okay, so that's why it's best. My opinion, it's best. In my opinion, it's always better to go to these specialized annotation tools because, well, I don't know if you remember one of my like that, for example, because it has been manually created and many people have been involved in the generation of video.
Okay.
Thank you very much. Yeah, the question is, well, we have been talking about magnitudes, displacement, and so on, but we didn't talk about NURAS. So how valid is NURAS? I think well, I never use NURAS. I know that it's a website, right? You can upload your metagenome and so on, and you will load FASTQ files or raw data, and then if it's very big, it will take weeks, I guess. Yes. Yeah, I don't have experience with NGRAPH. I'm very sorry. Maybe just you have experience in the past, a long time ago. Yeah. Yeah. So maybe they have a new version. Really, well, I don't know when was the latest update of NGRAPH. I know that it was released in 2008, 9. Yeah, 2008, 9. Yeah, and then that's if they update it, of course, it will be much better for sure. Yeah, but you have other options, the microbiome analysis. You can do all of these analysis as well. Are you familiar with that?
Also, Galaxy.
Yeah. Yeah. Eggnog, yeah. So she's asking why eggnog. I said that it was, well, it's just because it contains much more annotations. So it contains egg, it contains cough, it contains geo terms, Guillermo, tell me, which PFAM, yeah. Then you have so many different annotations that at the end you will reduce a lot of okay. Depends on your question. Okay? So depending what you want to know about your sample. But for example, for us, it's very useful. And I'm talking about my group and also your group. I think, yeah, core is one of the best databases because it's hierarchically classified, and then you can get different categories. So you can get patterns of functions. EPAM is quite nice because the annotation is, well, there are quite a lot of programs annotated there. So it's always, yeah, there are always advantages. And of course, it's more time consuming because EPAM will take maybe longer time than in Guillermo or it's HTML provide. Yes, the good thing, for example, of PIFA or Ivica versus Co or Care is that it's based in this HMM model. So then you are not just aligning your gene to a representative gene. You're aligning your gene to a model that is a consensus of multiple of the same genes from different bacteria. So also it allows you to get annotation further in biology. At the end, what you want is to have as many genes annotated as possible. Because if you get a hypothetical product, yeah, what do you do with that?
Okay, I think we can stop.
You said the capital frequency, you mentioned back in the
I shouldn't say.
yeah, but maybe I can do this before how do you need to make a prediction of function?
I saw that question coming. Yeah. Okay. Well, you have several tools. I said that with system amplicon and ITS and so on, you can only get taxonomic information. There is a but, and yeah, that's not entirely true. So you can do predictions because there are some programs or databases where they use genomic information that is already out there, and then they compile it in a database, and then you put your CCNS amplicon in that database. I never use it. It's called PyCraft. It's one. Tax for Fun. Shine as well. I think there are more. I never use it by myself. Well, the only thing is that you are basing your functional analysis in only 350 base pairs. So how accurate is that in terms of strain composition? I don't know. Yeah.
Yeah, there are two papers where they actually compare the outcome of these types of programs that infer functions from a phylogenetic marker. And let's say they did not come out so well, but they are getting better. But originally, they were developed for, I think, the human gut microbiome, where there was sort of like, I mean, it's a relatively small community as compared to environmental microbiomes, right? So there, I think the accuracy was better. But again, it's improving the more information you get, it improves. But we prefer to do the metagenomics.
So you mentioned that's quite a good point. So, PyCraft, I think it was designed mainly for the gut microbiome. I think. And Tax for Fun was more for environmental. So if you have to choose one, you are working with soil, I guess it should be in Tax for Fun, but I wouldn't, exclude the other ones. Yeah. So what has been your experience? Now it's my question. So did any one of you have experience with that? With PyCraft or Tax for Fun or any other? Is that
China. They're using China. But it gives different technology importance of that. It doesn't pinpoint particular function.
Yeah. Okay. Do you have more questions? Yeah.
Victor?
Lisa, question for you.
No, I just remembered that it would be interesting for all of you that you have that easy access to servers. This website that is called K-Base, and there you have what they call narrative. So there you have a lot of these tools that we talk about, and then people do like a small code there where you can upload your data and then follow the narrative and run their code in a really more interactive way in their servers. It's not a super fast option, but it's a really good option where you have most of programs. Kbase.us. I yes, is that one. So there you can log in and then you can search for some of these software that we talk about, and then you won't have to deal with installation, and it will be easy for you if you don't have access for a server. careful with who you are you sharing your data. Questions, remarks? No? Okay. Well, then coffee break? Okay.
Great.
Thank you, Kumar.
Yeah.
Let's have a quick coffee
So,
break. Then what?
yeah. So we will break for tea now. After tea, we are going to lab to see the plates. Okay. Then once that is done, we come back here for a few more announcements and a few more discussion points. We will do that. Okay.
So, Kumar, if there is time, I could show how to do work and maybe a bit of bioflam.
Yeah, we will do that.
By the way, there is bioflam 4. It just came out last week.
We will do that. After tea, just come back here and